photosynthesis can be divided into two reactions. We have a light-dependent reaction. The plant is getting energy from the sun. And we also have a light-independent reaction, and we'll be focusing on that today. This is called the Calvin cycle. The Calvin cycle takes place in the stroma inside the inner membrane of the chloroplast. The light-dependent reactions, you see the energy from them being stored, the energy and light being stored in a chemical form in ATP and NADPH. And these are used as energy in the Calvin cycle to fix carbon molecules from carbon dioxide or CO2 into energy-rich sugars. The function of the Calvin cycle is to produce glyceraldehyde 3-phosphate or phosphoglyceraldehyde, also known as G3P or PGAL. We'll refer to it as PGAL. So PGAL are 3 carbon carbohydrates that can be used to make other more complex sugars like glucose. And glucose is a 6 carbon molecule made from 2 PGAL. So we need 3 turns of the Calvin cycle to produce enough PGAL to see one of these transported into the cytosol of the cell. The rest of the PGAL are used to regenerate the components necessary to run the cycle, as I will describe in a moment. Let's dive into more detail about the Calvin cycle. We can break it down into 3 phases. We have carbon fixation, reduction, and regeneration. The carbon fixation phase, as we said, fixes carbon into organic molecules. So CO2 entering from the stomata combine with a 5 carbon molecule called ribulose 1,5-bisphosphate, or RUBP. This reaction is catalyzed by the enzyme ribulose 1,5-bisphosphate carboxylase oxygenase, also known as Rubisco. H2O hydrolyzes the 6 carbon intermediate molecule, 6 because we have 5 and 1 here to make 6, to make 2 3 carbon molecules called 3-phosphoglycerate or 3-PGA. Now we enter the reduction phase. This phase requires much of the ATP and all of the NADPH used in the Calvin cycle and reduces the product of carbon fixation to glyceraldehyde 3-phosphate or PGAL, most of which continues in the cycle. To get there, 3-PGA from the fixation phase is phosphorylated by 3-PGA kinase. This uses 2 ATP per CO2 fixed, and we get a 3 carbon intermediate molecule that is reduced with a dehydrogenase using 2 NADPH per CO2 fixed to form glyceraldehyde 3-phosphate or PGAL. Finally, we enter the regeneration phase, which is complicated with many stages and enzymes to reconfigure the carbon molecules to ribulose 5-phosphate, which the plant phosphorylates into RUBP using ATP, and this begins the cycle all over again. All carbon in the Calvin cycle is conserved, so this rearranging of carbon molecules just works to make RUBP. Remember that the regeneration phase is using the 3 carbon PGAL molecules from the reduction phase. I'll talk generally about the rearranging happening here. In this phase, PGAL combines with other PGAL molecules to form a 6 and then a 9 carbon molecule. And this gets catalyzed into a 5 and a 4 carbon molecule. This 5 carbon molecule needs an enzymatic reaction to create the ribulose 5-phosphate that will get phosphorylated into RUBP. So we'll say that has happened once, and we have 1 of the ribulose 5-phosphate here. We still have a 4 carbon molecule, and this is able to combine with more PGAL to get a 7 and then a 10 carbon molecule. And then this is broken down into 2 5 carbon molecules Let's pull that down. We have two 5-carbon molecules, and these are reshuffled again to become the 5-carbon precursor right here for RUBP. So at this point, we have 3 ribulose 5-phosphate molecules, and to get these to RUBP here, another ATP is used per each of these 3 molecules to phosphorylate ribulose 5-phosphate into RUBP. So in summary, 3 revolutions of the Calvin cycle takes 9 ATP and 6 NADPH. This comes from the light-dependent reaction, and we fix 3 CO2 molecules to produce 1 3-carbon carbohydrate. And don't forget that a big part of the Calvin cycle is regenerating RUBP to continue the cycle again. Note that to get 1 carbohydrate like glucose, which is a 6-carbon molecule, the plant would need to perform double that. So 6 revolutions of the Calvin cycle. And it would require double the energy that I mentioned here. Thank you for watching, and I hope my video has helped you better understand the Calvin cycle and its importance.